Hello, my name is Angelica Wong, and I will be talking about my fall 2016 Bio 171 lab sessions, which focus on escape pathogens and antibiotics. The overall objective of the sessions was to collect soil samples that could be brought back for testing so that microbiological methods could be done on bacterial colonies that could be possible antibacterial producers. With the bacterial growth, colonies on cultures that were isolated would be studied, and these colonies would come from a single cell. Based on the area in which the soil sample was found and the surrounding environment that it was within, it should be possible to extract a sample rich in microbes that could be taken to the lab and find a method to isolate single colonies of bacteria. To start off, I'd like to introduce some topics that relate to this lab, such as what pathogens are, as well as escape pathogens, and then I would also like to discuss their discovery. Pathogens are disease-causing microorganisms. They were discovered by German physician and bacteriologist Robert Koch. While performing experiments to determine the agents responsible for tuberculosis and anthrax, Koch established a series of postulates that could be applied to identify pathogens responsible for other infectious diseases. His postulates include the following. The microbe is always present in diseased individuals but not healthy individuals. The microbe is isolated from the infected individual and grown in pure culture. The microbe in the pure culture is introduced into a healthy individual and the same disease occurs. The microbe is re-isolated and shown to be the same as the original. This process might seem second nature in current day, but in the late 1800s, it was revolutionary. Escape pathogens are a collection of microbes that are considered to be more of a threat throughout the healthcare setting, since they contribute to the predominance of the emerging antibiotic resistance present in organisms. These pathogens have different abilities to resist antibiotics, such as mutation, enzyme deactivation, gene acquisition, and prevention of drug accessibility to its host. They are some of the most important microbes to study due to the fact that they disrupt the conventional physiology relevant to humans. With the low rate of drug development within current generations, escape pathogens have taken a central role as new drugs are desperately needed to counteract them. Due to their dangerous properties and possibility to influence organismal mutations that can cause further problems within human physiology, non-pathogenic variants are some great relatives for further studies and tests with regard to their overall similarities. Here is a list of the escape pathogens along with their safe relative. The highlighted in orange are the ones chosen to conduct the lab for the semester. The chosen escape pathogens, or actually their safe relatives that I did research with, are S. epidermidis, and E. coli. S. epidermidis is a gram-positive pathogen. The natural environment of it is the human body, and it usually originates from disease. Since the bacteria usually lives on the skin and hairs of all human beings and is a nosocomial pathogen, it is important to be able to identify the specific strains it has. E. coli, on the other hand, is a gram-negative pathogen. Most E. coli strains do not cause disease, but virulent strains can cause gastroenteritis, urinary tract infections, and neonatal meningitis. It can also be categorized by severe abdominal cramps, diarrhea that typically turns bloody within 24 hours, and sometimes fever. It is important to remember that not all bacteria are harmful, as a lot of them can help within the proper regulation of bodily systems. Some reside in human bodies and even outnumber cells. Additionally, these microbes also benefit health, as showcased by those that fall under the category of probiotics. The discovery of antibiotics was a milestone within the medical world, most notably applicable to its importance in biological research. For example, the discovery of penicillin by Alexander Fleming catapulted the motivation towards the quest for antibiotics. Prior to penicillin, highly corrosive antiseptics were utilized to treat severe infections damaging human tissues and the immune system. In antibiotics, structure proves to be an important trait shared among different classes, since it modifies the overall function. As an example, the classes of B-lactams antibiotics share a 4-meter ring structure, allowing them to inhibit bacteria cell wall synthesis. Their mechanism of action varies by their molecular target, arguably concerning their specific biomechanical interactions, contributing to their inhibitory effectiveness towards specific bacteria. This contributes to the theory that antibiotics are selective, thoroughly implying that they focus on certain cellular particles specific to given bacterium. For the span of 1 week, various lab procedures were done to find any possible candidates for antibiotic properties in soil microbes. And here's the hypothesis that I came up with. Hypothetically, if unknown bacteria samples are patched onto agar plates containing escape pathogens, then it is possible to find antibiotic-producing microbes based on their zone of inhibition, because the synthetic tailoring of antibiotics allows them to have specific strengths or properties to kill more bacteria. So, within the span of 1 week, there were various tests done for the lab. The first test done was patch plating. Once serial dilution was complete, bacterial samples were patched onto an agar plate using a grid system. This was done by scraping off small samples with a toothpick of what was believed to be possible antibiotic producers from certain plates of the dilution series. These were transferred to a new plate that had a drawn grid system that was numbered to help with future referencing. A different sterile toothpick was used for each sample taken. Once this process was done, the plate was sealed with parafilm and incubated for about 2 to 3 days. After the incubation period, the results from the patch growth were tested against two chosen safe relatives of escape pathogens. As mentioned before, the chosen pathogens were S. epidermidis and E. coli. To start, 1 ml of LB broth was placed in a centrifuge tube and the chosen escape pathogens were resuspended. Resuspension was done by using an inoculating loop and scraping up a small quantitative amount of the safe relatives. These pathogens were carefully inoculated in the liquid broth. Then the tube was capped and vortexed so that any possible chunks could be removed and mixed thoroughly in the broth. This process was done one time for each escape pathogen. Using a pipette, 150 microliters of each suspended pathogen was placed onto 10% and 50% TSA plates that had drawn grid systems on them, totaling 4 plates with the pathogens. Samples from the initial patch work were repatched onto the 4 plates using the patch method described previously and incubated for a little over 24 hours. Patches number 2, number 3, and number 6 were chosen to be used for the remaining tests to follow. For catalase testing, a clean microscope slide was used and sectioned off into thirds. Each section had 1 to 2 drops of 3% hydrogen peroxide solution placed. Using a sterile toothpick, an isolate colony from the most recent patchwork was picked and scraped. Then the end with the picked cells was directly placed into the solution and bubble formation was observed. This was done 2 times, the first showing the positive negative controls along with the number 2 patch. The second one showing the 3 bacterial isolates. For glucose fermentation testing, a sterile inoculating loop was used to scrape a bit of the number 2 patch. It was carefully inoculated in a test tube that had liquid glucose fermentation media with phenol red indicator. This was done 2 more times for number 3 patch and number 6 patch, each done separately. The 3 tubes were capped and incubated in a shaking incubator for about 48 hours. To test for any motility, the end of a sterile inoculating needle was wiped with an alcohol wipe and air dried while being very careful to not allow it to touch anything. The needle was used to swipe the number 2 patch with a quantitative amount and then stabbed carefully through the center of a test tube containing motility media. This procedure was done 2 more separate times for patch number 3 and number 6. The test tubes were then capped again and incubated in a shaking incubator for about 48 hours. For the procedure of liquid culture growth, a sterile inoculating loop was used to scrape a bit of the number 2 patch. It was carefully inoculated in a test tube that had liquid TSA culture. This was done 2 separate more times for the number 3 patch and number 6 patch. The 3 test tubes were then incubated in a shaking incubator for 48 hours as well. For gram staining, a clean microscope slide was obtained and a hot plate was heated to 80 degrees Celsius. 10 microliters of distilled water was pipetted evenly across one end of the slide, and using an inoculating loop that had a small bit of number 2 patch, it was placed into the water droplet and the bacterium was suspended on the slide. This was done 2 more times, each separate, for the number 3 patch and number 6. The slides were allowed to air dry and then heat fixed by carefully placing the ends with the bacteria onto the hot plate for about 15 minutes. After heating, the fixed microslides were placed onto a staining rack with a staining tray underneath. The following described protocol was done for each slide, one at a time, to ensure quality staining was done for each. First, crystal violet was evenly flooded onto the slide on the end with the bacterium, with a wait time of 1 minute for proper gram-positive detection. After the minute, the excess crystal violet liquid was gently washed with distilled water. After proper washing, gram iodine solution was flooded and allowed to stay for 1 minute to keep the crystal violet from being removed. It was then gently rinsed with distilled water again, after which the slide was rinsed with decolorizer solution for 10 seconds and or until the solution ran clear so that it washed out the purple complex for bacteria with thin cell walls. It was then rinsed with distilled water for the proper application of the next solution. Lastly, safranin statin was gently flooded onto the slide for a minute to act as the counter stain, turning gram-negative bacteria into a hue of pink-red, after which the excess safranin was washed out with distilled water. The slide was then left to air dry after the protocol process. After drying, the slides were observed under oil microscopy for gram classification in regard to isolated colonies. For PCR testing and gel electrophoresis, the following was done for each of the chosen bacteria candidates. Totaling 6 microfuge tubes, the first set of tubes, all labeled 16S, contained a PCR bead. Then these liquids were added to the tube in order as mentioned: 10 microliters of water, 10 microliters of 16S primers, and 5 microliters of DNA. The second set of tubes, all labeled Uni for universal, contained a PCR bead, and the following liquids were added to the tubes in order as mentioned: 10 microliters of water, 10 microliters of universal primers, and 5 microliters of DNA. These 6 prepared tubes were placed into a PCR machine for about 48 hours. The 6 tubes were then spun some more using a mini centrifuge machine for about 1 to 2 minutes. 7 microfuge tubes, each containing 3 microliters of UV View blue dye, had 15 microliters of each of the 6 PCR liquids transferred singularly to the tubes, 1 PCR liquid per tube. The 7th tube had 15 microliters of weight marker added. These 7 samples were then placed very carefully in a horizontal agar DNA gel electrophoresis apparatus for about 45 to 60 minutes. The gel that resulted was then placed into a machine for result viewing. Post-lab, the stains of the candidate bacteria were analyzed using a microscope and camera. Digital pictures were taken of the selected sections of the stain, and after machine processing of the gel, a digital image was also taken of the results. The results of the lab work I did are as follow. For patch plating, the circled and numbered colonies were the ones selected for patch plating and testing against escape pathogens. And to the left is the final growth. When testing against escape pathogens, patches number 2, number 3, and number 6 were used for the durations of the tests conducted for the remainder of the lab sessions due to showing zones of inhibition, meaning that they could be possible antibiotic producing microbes. For catalase testing, all 3 candidates showed to be positive in producing catalase. Patch number 2 rapidly bubbled, patch number 3 simply bubbled, and patch number 6 simply bubbled as well, but it dissipated a lot faster than the other 2 patches. For glucose fermentation testing, all 3 fermentated except for bacterium number 2. Number 2 turned the glucose fermentation media a bit pink from red, which made it a bit difficult to determine precise results, while number 3 and number 6 turned the glucose fermentation media yellow from red, indicating acid production and that they can use glucose for carbon and energy. For the motility assay, none of the candidates showed motility, meaning that each bacterium cannot quote-unquote swim by means of flagella. Liquid culture growth showed that all candidates had some sort of growth, but in the picture, it cannot be seen very clearly, but number 3 was more translucent with a little ball-like clump at the bottom. Gram staining showed that they were overall gram negative and shared the characteristic of being coccus. Patch number 2 had a chain arrangement, while patch number 3 were in forms of clusters. Patch number 6, however, showed some sort of symbiotic-like relationship due to the presence of both gram-negative and gram-positive bacterial cells, as indicated by the mix of dark purple and pink throughout. Several reasons for this happening could be improper isolation of the bacteria, improper gram staining protocol done, or a true symbiotic relationship was showcased. For PCR and gel electrophoresis, The gel electrophoresis bands indicated bacteria and nanograms. 3 of the 6 lanes had successful readings. Lanes 1, 3, and 4 were 100 BP in size. To conclude, here is what I have to say about my overall lab work. When testing against the escape pathogens, there were 3 viable candidates for antibiotic-producing microbes found. True to my hypothesis, viable candidates were found based on having zones of inhibition. However, this was only seen on the 10% TSA plate with S. epidermidis, thus meaning its absence in the other plates could mean that, conditionally speaking, some sort of contamination or even environmental factors inhibited its growth in fighting back the other sample of S. epidermidis and E. coli. And in the future, here are some things that I would possibly do to do any further testing. To determine whether or not these possible candidates of antibiotics that I found are actually truly viable, further testing would need to be conducted. This could be done by sending the result of the gel electrophoresis for DNA sequencing. So, if studies and investigations on microbes found in soil continue to be done like what was done the entire semester based on the Small World Initiative procedures, in time, new antibiotic producers may be found and developed for use to end the antibiotic crisis. To wrap things up, here are the references I use for my research. Thank you for watching.